So we are starting the streaming now in 3,
Perfect.
2, 1. You are online.
So welcome to the global talk session on open source vaccine development, practices, opportunities, and tensions. My name is Milena Leybold, and I present a working paper that I work on together with my colleague Konstantin Hondros. And the paper has the title Repurposing and Tinkering, How Open Source Vaccine Initiatives Alternatively Organize for Novelty. I would like to start my presentation with a quote by Peter Meyer-Bardruck, who also contributed to this conference yesterday, I think. He said in May, perhaps even crueler than hoarding vaccine doses is hoarding knowledge of medical technology that can end the pandemic. And building upon his quote, I would like to emphasize that the innovation that was brought forward by biotech and pharmaceutical companies during the pandemic was exceptional. However, still many countries lack access to vaccines. And public debates arose on alternative ways of handling IP in vaccine R&D beyond the petrified standards of the Western-dominated pharmaceutical industry. They addressed the potential of open source that could tackle both challenges during pandemics, namely accessibility and innovation. So drawing upon literature of the field of organization studies where Konstantin and I come from, we problematize and try to grasp potential tensions that initiatives aiming for such alternative approaches to IP face. So I outline the theoretical framework that we build upon. It's the literature on organizational isomorphism, and we link that literature with literature about the emergence of novelty in organizations. Isomorphism is the phenomenon that organizations become increasingly homogeneous despite and while aiming for change. Such change that would be, for example, required to solve the challenges of pandemics. And this isomorphism is due to their tendency to reduce uncertainty and gain legitimacy by following standards, norms, and by doing as everyone does. And we say that the westernized pharmaceutical industry, mainly in response to regulative standards, has developed highly isomorph organizational practices that might make the entering of outsiders quite difficult. So novelty still emerges in this isomorph field, that's without doubt. However, actors who might not be part of this field yet, for example, because of applying alternative approaches to vaccine R&D, might carry important sources or important novelty too. So we find it puzzling how alternative ways of fostering novelty can be embraced and can gain legitimacy in organizational fields that deeply rely on isomorphism. And to scrutinize this puzzle, we ask the following question: how do open source vaccine R&D initiatives alternatively organize for novelty? And to answer our research question, we compare two empirical cases that both build on an open source practice. And since we are particularly interested in processes, practices, and potential uncertainties that these initiatives deal with, we collected qualitative materials such as meeting observations, interviews, but also white papers that they publish. And we try to draw processual conclusions and set them in context with developments of the SARS-CoV-2 crisis. So I briefly introduce the two cases that we work with. The first one is Vaccinum. It was formerly called OpenVax. It's supported by the Open Source Pharma Foundation, was founded 2020 as in response to the COVID crisis, And its initial focus was on finding an open source COVID-19 vaccine. And it's now aiming for discovering an ultra broad spectrum open source vaccine through processes of repurposing of already existing, widely available, approved, licensed, widely accepted, non-exclusively manufactured, off-patent, live-attenuated vaccines that have been shown to elicit trained innate immunity and may provide broad spectrum protection. The advantage of doing so, as they do so, of repurposing, is that you can skip clinical trials until phase 3, and that you have a high accessibility to the vaccines you want to work with. We compare this case to REDVAC. It's a citizen scientist initiative funded in direct response to the COVID-19 crisis. They were also focused on developing a vaccine before they become commercially available. So it was rather the focus on bridging the time until another vaccine is on market. And with the commercial vaccine success came a change in their self-description of REDVAC. So they are now challenging industry vaccine R&D as they want to provide a more rapid, a more accessible and globally oriented vaccine R&D alternative by developing a vaccine platform that relies on the multiple and scattered tinkering of citizen scientists throughout the world. Their peptide-based vaccine is administered intranasal, and the platform and the trial designs are under a Creative Commons license. So you can access the white papers of REDVAC also on their website. The advantages of such an approach is that it's transparent, for example, that's among other advantages, and there's room for own development. And we call that practice that they apply to organize for novelty tinkering. Okay, so what do we see here? Both initiatives don't rely on traditional IP to protect their R&D. They rather build on knowledge sharing, openly knowledge sharing. And however, their practices differ while facing similar challenges and situations during the COVID pandemic. So we found these two practices of open source R&D that we see as practices applied to handle the tensions between meeting the need for legitimacy and similarity on the one hand, because they can't do something completely different and need to follow some standards. And on the other hand, providing an alternative for organizing novelty that is sustainable and accessible. The initiatives apply practices that are as different as possible and as similar as needed for coping with uncertainties and gaining legitimacy in an isomorph field. When repurposing, they apply already existing off-patent vaccines and use it for different contexts. The outcome of this application is not protectable. So the invention is not protectable anymore. And in the case of tinkering, this is different. It allows for multiple and parallel incremental variations of the initial vaccine. So the incremental changes are not protectable themselves. However, there could be room for commercialization when variants are modified enough. Comparing both, we would argue that the source of legitimacy and uncertainty reduction in repurposing lies in the use of something that is already approved. And in case of tinkering, it could be the outlook to rather traditional commercialization that might be a source for legitimacy in the field. And we discuss in our paper that we would not only limit these two practices to their work on vaccine itself, but rather to their whole organizational identity that is repurposed in the case of Vaccinome, so from OpenVax to Vaccinome, from COVID only to ultra broad spectrum, and tinkered in the case of RADVAC from bridging time until a COVID vaccine is available to step-by-step providing and improving a vaccine platform that could be leveraged also for future pandemics, as well as providing a new testing framework, challenging the field standards more actively. So we see two initiatives that both face tensions between being similar and being alternative to traditional vaccine R&D. And building upon our investigations, we developed some practical implications that I will outline now to conclude my presentation. We asked how can we embrace the alternative part of such initiatives. So the first point is large and easy to use patent databases, for example, could help to reduce uncertainties, particularly regulatory uncertainties. Initiatives can then concentrate a little bit more on their R&D work. This could also be supported by legal advice to help such initiatives coping with potential IP barriers. Further, funding is always an issue for all vaccine R&D efforts, but for initiatives that alternatively organize for novelty, it's particularly difficult to get their trials funded. So providing new sources, how these initiatives could get funding for trials would be another important step towards a more diversified field of vaccine R&D. And last but not least, we all as individuals could help them to increase their legitimacy by sharing information about them and also, for example, helping to establish contacts for collaborations. So that's all from my side. Thank you very much for listening and for having me. These are the sources I've used in the presentation. And if you have any comments, questions, ideas, I'm very happy to hear them, or we are very happy to hear them and answer them via email. You should see the email addresses listed there, Konstantin Hondros and from myself. Thank you very much for your attention, and I hope to see you soon during the conference.